Hey everyone, and welcome to RUBIN'S Mind Lab, where we break down the latest research into what it really means. Ever wonder why studies you read about seem to contradict each other, or why a finding that seemed solid just a few years ago now seems shaky? Well, this new research dives deep into that very question. Imagine you are trying to understand how a group of people behave, so you measure them all at one point in time, you average their responses. Sounds sensible, right? But what if each person is on their own unique journey, with ups and downs and twists and turns that are totally individual? This new paper argues that by just looking at averages, we're missing a huge part of the picture. And it is why some research findings just don't hold up when others try to repeat them. You've probably heard about the replication crisis. It is this idea that many scientific findings, especially in psychology and social sciences, aren't as solid as we thought. When researchers try to repeat a study, they often don't get the same results. For a long time, we've blamed things like not having enough participants, secrets about how experiments were run, or just looking too hard for positive results. These are all important. And the paper mentions them, referring to them as methodological flaws like low power and publication bias. But this new research says there is something deeper, a fundamental issue that these typical explanations miss. They say that human behavior and psychological processes are nearly always what they call non-ergodic. So, what exactly is non-ergodic? Think of it like this. An ergodic process is one where if you watch a lot of people for a short time, you learn the same thing as if you watched just one person for a really long time. It means that what happens on average across a group tells you a lot about any single person. But human lives aren't like that. Our emotions, our thoughts, our actions, they change from day to day, even hour to hour. And importantly, every single person has their own unique trajectory. So if you take a snapshot of a group, the average might not actually describe anyone in that group very well. Imagine trying to understand a symphony orchestra by just listening to the average volume of all instruments at one moment. You wouldn't really grasp the music, would you? This is where the problem of Estimand drift comes in, because people are so different and change over time. What a study finds depends heavily on who is in the sample and when the measurements were taken. If you have a group where half the people are feeling really happy and the other half are feeling really sad, the average might be neutral. But if you pick a different group where mostly happy people are present, your average will be high. If your next study, a replication, happens to grab more sad people, your average will be low. The thing you are trying to measure, the Estimand, has drifted because your samples are different. The researchers point to studies that show how much individuals vary compared to how much groups vary. For example, one study found that the ups and downs within a single person's life were 2 to 4 times bigger than the differences between people. Another example they give is about values. On average, acting on your values predicts feeling better, but some people, like a group called Stoics in one study, actually showed the opposite effect within themselves. The average told us almost nothing about these individuals. This is the core of the problem. If you have a lot of this sign heterogeneity, meaning some people show an effect in one direction and others show it in the opposite direction, then your average will depend entirely on the mix of people in your study. And when researchers use convenience samples, which are just groups of people who are easy to access, the mix of people can vary a lot from study to study, and especially from an original study to its replication. Now, here's where it gets mind-blowing. The authors present a mathematical argument. They say that even if a study is perfectly designed, has enough participants, and is conducted with absolute honesty, there is a guaranteed minimum rate of replication failure. This is the structural floor they talk about. It's not due to sloppiness, it is a mathematical consequence of human complexity. Let's break down the math they introduce. They imagine a situation where a fraction of people have a positive effect and another fraction have a negative effect. Let's call the fraction with the minority sign effect. Q, the ensemble mean, is the average effect across everyone. If Q is close to half, the ensemble mean can be positive. But it is hiding a lot of opposing individual experiences. They then model how convenience sampling works. This means that each study just happens to get a certain mix of people. If a study gets a mix where less than half the people have the minority effect, it will likely find a positive result. But if the next study with a different random mix gets a mix where more than half have the minority effect, it will find a negative result. Even if the overall population has a positive average, this leads to a calculated failure floor. This equation shows that how often studies fail to replicate is determined by two things researchers rarely measure. Q, the actual proportion of people experiencing the effect in the opposite direction of the average, and which measures how much the composition of samples tends to vary. Importantly, They state that simply increasing the sample size won't fix this. A bigger sample just makes you more certain about the average of that specific kind of sample, which might still be different from the average of another sample. The paper then shows how this floor plays out. They have figures illustrating this. Figure 1b shows how this failure rate changes depending on how much sign heterogeneity there is and how much convenience sampling is involved. It is a surface independent of sample size. They estimate that for a process with moderate differences and moderate convenience sampling, the replication success rate could plateau at around 68%. This means 1 in 3 studies could fail to replicate even if they are perfectly done. This is a structural limit. They even connect this to real-world replication crisis numbers. For a specific scenario they model where the minority sign effect is quite large, around 42% of people, and sampling is convenient, the perfect study floor is about 32%. When you add in the usual limitations of real studies like not perfectly powered samples, the expected failure rate jumps to about 52%. This matches the roughly 51% failure rate observed in a major replication project. So, a significant chunk of what looks like bad science might actually be the inevitable outcome of human complexity. So, what is the solution? The paper argues we need to shift our focus from just looking at group averages to understanding the idionomic distribution. This means measuring psychological processes longitudinally, meaning over time within each individual. They call for an idionomic approach. This involves collecting a lot of data from individuals over time. By doing this, you can estimate the effect of a predictor on an outcome for each person separately. Then, you can look at the distribution of these within-person effects across people. This distribution would tell us about the minority, sign mass Q, and how much people vary. This approach allows researchers to distinguish between true methodological failures, like low power or biases, and the mathematical consequences of human complexity. If we know the idionomic distribution, we can predict how often studies should replicate, and if they fail more often than predicted, then we can look for methodological problems. They introduce tools like the Idionomics R package that can help with this kind of analysis. The key takeaway here is that we need to move from a cross-person ensemble average, which tells us about groups, to an idionomic approach which understands individuals first and then generalizes. Looking back at figure 1b, it really drives home the point that this floor is a structural issue. It is not about bigger sample sizes fixing everything. While replication success might increase with sample size, it hits a ceiling, a limit imposed by the very nature of the processes being studied. The paper is packed with these technical details and references to sophisticated statistical tools developed by the authors and others like IREMAX and TS Peruta, which are designed to analyze individual time series data. Table 1 provides an excellent overview of these idionomic methods, showing how we can move from understanding individual processes to grouping similar individuals. Now, what is my take on this? I think this paper presents a really compelling argument. It is a breath of fresh air to hear researchers acknowledge that maybe, just maybe, a lot of what we're seeing as a crisis isn't because everyone's doing bad science, but because the human mind and behavior are incredibly complex and variable. The idea of a structural floor on replicability is, frankly, a bit scary but also very logical. They're saying that some level of replication failure is mathematically guaranteed if we stick to traditional averaging methods when dealing with non-ergodic processes. What I find particularly interesting is how they propose a solution. The idionomic approach. By focusing on within-person, longitudinal data, we can get a much clearer picture of individual differences and how processes unfold over time. This seems crucial for truly personalized approaches to understanding and intervening in human behavior. It's like moving from a one-size-fits-all medicine to tailor-made treatments. Of course, this approach has challenges. Collecting intensive longitudinal data is demanding for both researchers and participants, and the statistical models needed can be quite complex. Though the paper does point to growing toolkits to help with this, but the implications are huge. If this is true, then a lot of past research might need to be re-evaluated, not as flawed, but as representing averages of highly variable phenomena. Future research needs to embrace these idionomic methods. We need to see more studies that report the distribution of within-person effects, not just the average. This will allow us to understand the structural floor and then have a better chance of identifying and fixing actual methodological issues. It's a call for a more nuanced, more individual-focused science. What do you all think? Does this idea of a guaranteed replication floor make sense to you? Let me know in the comments below. And if you found this breakdown helpful, please give this video a like and subscribe to RUBIN'S Mind Lab for more explorations into the fascinating world of psychology research. We'll see you next time.